Oxytocin, also known as the love hormone, is an incredibly versatile molecule, small in size but tremendously important in both physiology and behavior. It is a hypothalamic neuropeptide and was initially discovered back in 1906 for its ability to induce uterine contractions during childbirth. Over the decades, scientists realized that this 9-amino acid peptide functions not just as a hormone that travels through the bloodstream, but also as a neurotransmitter in the brain, influencing a wide range of processes. One of the reasons oxytocin has attracted so much attention is its remarkable versatility. How exactly does a single molecule carry out so many roles? The answer depends on factors such as where it acts, how much of the neuropeptide is released, what stage of development the individual is in, and the specific binding properties of the oxytocin receptor. In short, the effects of oxytocin can look very different in different contexts. From an evolutionary standpoint, oxytocin is highly conserved. We see homologous forms of oxytocin across a variety of species, including both vertebrates and invertebrates. In mammals, oxytocin is produced in the hypothalamus, primarily within 3 nuclei: the paraventricular nucleus, the supraoptic nucleus, and accessory nuclei. Within these nuclei, we can identify 2 main types of oxytocin neurons: magnocellular oxytocin neurons and parvocellular oxytocin neurons. Magnocellular oxytocin neurons project to the posterior pituitary, where oxytocin is released into the circulation. This is how oxytocin exerts its hormonal actions on peripheral tissues, such as the uterus or mammary glands. At the same time, magnocellular oxytocin neurons send signals to various forebrain regions, influencing behaviors and emotions. Parvocellular oxytocin neurons, on the other hand, are fewer in number and are found exclusively in the PVN. These neurons project to the midbrain, brainstem, and spinal cord. A particularly notable role of parvocellular neurons is their ability to coordinate or orchestrate the activity of the magnocellular neurons. While there is only one known type of oxytocin receptor, a G-protein-coupled receptor with high affinity for oxytocin, its presence in different tissues and brain regions allows for a wide variety of physiological and behavioral effects. These functions range from uterine contractions and lactation to modulation of social behaviors, memory, and stress responses. Indeed, projections from hypothalamic oxytocin neurons to regions such as the amygdala, hippocampus, and prefrontal cortex demonstrate how it can modulate emotional states, memory processing, and social bonding. Social interaction is more than just conversation or visual cues. It often involves physical contact and the integration of sensory inputs with emotional and motivational states. Many forms of touch, such as hugging, stroking, or caressing, can directly influence oxytocin release, which enhances bonding, reduces stress, and influences behaviors like parenting and mating. Research has shown that the nature of the touch matters. For instance, tactile contact that is perceived as affectionate or nurturing can prompt a different, often stronger, neuronal response compared to neutral or object-based touch. One interesting example from human studies is that heterosexual men perceive a woman's gentle caress as more affectionate than an equivalent touch by a male stranger, even if the actual touch was unbeknownst to them, performed by the same person. Various somatosensory stimuli relevant in social situations have been shown to modulate the oxytocin system. There are 2 major biological events where oxytocin's role is highly prominent: labor and breastfeeding. During labor, the physical stretching of the cervix and uterus sends signals through sensory nerves to oxytocin neurons in the brain. This triggers the release of oxytocin, which then travels peripherally to intensify uterine contractions. It's a feed-forward mechanism. The more the uterus is stretched, the more oxytocin is released, and the more contractions are amplified. In breastfeeding, a very similar pathway applies. Sensory stimulation from the infant suckling at the nipple is conveyed through C-type sensory fibers to the spinal cord. eventually activating oxytocin neurons in both the paraventricular and supraoptic nuclei. This, in turn, causes oxytocin to be released into the bloodstream, inducing the milk ejection reflex. After a vaginal birth, both mother and infant show elevated levels of oxytocin, and skin-to-skin contact immediately after birth has been reported to further boost oxytocin levels. This additional increase encourages newborn behaviors like spontaneous breast-seeking behavior and also leads to more vocalization and less crying. Interestingly, after a cesarean section, the usual immediate surge in maternal oxytocin is often missing. Even skin-to-skin contact or initial attempts at suckling might not produce the same spike in oxytocin, presumably because the typical sensory feedback loop from labor is absent. Beyond these life-stage-specific events, gentler social touches, such as stroking, hugging, or caressing, can also release oxytocin. In many instances, both parties involved show elevated oxytocin levels during friendly interaction. Rodent research has demonstrated that even 10 minutes of stroking can raise plasma oxytocin levels in the animals, enhancing affiliative behaviors. In humans, forms of contact like massage or hugging can increase oxytocin levels, although the degree to which this happens varies widely from person to person and can depend on the study's design. Research points to the specialized role of C-tactile afferent fibers in the skin. These fibers respond best to soft, slow stroking at around skin temperature, precisely the kind of touch that is often interpreted as pleasant or comforting. These signals travel to the nucleus tractus solitarius, which then connects to the oxytocin neurons in the PVN. This creates a feed-forward loop. A gentle, comforting touch leads to the release of oxytocin, which reduces stress and reinforces bonding. Our study published in 2020 sheds new light on how specific oxytocin neurons in the paraventricular nucleus respond to social touch. The approach used to record the activity of oxytocin neurons in the PVN utilizes a novel optotetrode technique in combination with viral expression of light-sensitive ion channels. A recombinant adeno-associated virus was injected into the PVN of rats, thereby expressing channel rhodopsin under the control of the oxytocin promoter. Simultaneously, optotetrodes, which are optic fibers coupled with recording tetrodes, were implanted above the PVN. After blue light activation, neurons that reacted instantaneously with increased activity to this optogenetic stimulation could therefore be identified as oxytocin neurons, and were later recorded with the tetrodes during several socially relevant behavioral tasks. Intriguingly, these identified oxytocin neurons fired at a low baseline rate when the rat was alone. But their firing rate increased dramatically when the rat freely interacted with another rat. Crucially, this effect disappeared if the partner was behind a transparent barrier. So, just seeing, hearing, or smelling another rat wasn't enough to boost oxytocin neuron firing. Direct physical interaction was required. We could also show that this effect could be mimicked even in anesthetized rats by applying air puffs to the dorsal body region, indicating that the somatosensory component itself is key for oxytocin neuron activation. A particularly fascinating discovery was that parvocellular, but not magnocellular, oxytocin neurons seem to be activated by this sensory input. When we decreased the activity of parvocellular oxytocin neurons using a chemogenetic technique, the overall response of all PVN oxytocin neurons to touch was also reduced. This suggests that parvocellular oxytocin neurons function as first responders to somatosensory inputs and directly synapse onto the population of magnocellular oxytocin neurons to relay the information of touch. Additionally, these parvocellular oxytocin neurons do not only react to somatosensory stimuli, but their activity also directly influences social behavior. Activating or silencing these cells either increased or decreased social interaction, but again, only when the rats could physically interact with one another. If the partner was stuck behind a clear wall with no possibility of touch, altering parvocellular oxytocin neuron activity made no difference. After inhibition of parvocellular oxytocin neurons, the rats even started avoiding tactile contact from their cagemate, such as climbing or being climbed on. So far we have shown that oxytocin neurons are influenced by touch. But the interaction between somatosensory stimuli and oxytocin activity is actually bidirectional. Oxytocin also modulates how we perceive somatosensory stimuli, particularly painful stimuli. A number of studies in both animals and humans have explored this aspect, generally revealing an analgesic effect of oxytocin. In animal studies, there's substantial evidence that oxytocin reduces the perception of acute pain triggered by electrical, thermal, mechanical, or chemical stimuli. Out of 33 animal studies surveyed, 29 reported a reliable analgesic effect of oxytocin. Human studies also show a pain-reducing effect, but the results are less consistent, likely due to differences in study design, route of oxytocin administration, and individual variability among participants. Nonetheless, there is a clear picture of where and how oxytocin exerts its pain-modulating influence in the central nervous system. Oxytocin innervation and oxytocin receptor expression are found in key regions of the ascending and descending pain pathways, including the spinal cord, the periaqueductal gray, and the amygdala. By acting on these areas, oxytocin can dampen nociceptive processes at multiple levels. Interestingly, rodent studies suggest that there is a dedicated circuit for this, orchestrated by a small group of parvocellular oxytocin neurons that project onto the magnocellular neurons and help regulate their activity. Very similar to the processing of social touch that was shown earlier. Pain relief can be achieved via two main routes. First, peripheral release of oxytocin through the pituitary into the bloodstream, and second, direct spinal cord inhibition of nociceptive neurons. The current hypothesis suggests that magnocellular oxytocin neurons primarily address the emotional and cognitive aspects of pain in the forebrain, for example, how stressful or upsetting the pain feels, while parvocellular oxytocin neurons drive the more robust analgesic effects on peripheral and spinal levels. This dual effect, reducing the stress associated with pain while also diminishing the pain signals themselves, makes oxytocin an interesting candidate for therapeutic approaches targeting chronic or acute pain. In summary, oxytocin sits at the intersection of sensory input and social behavior. On the one hand, social touch and other sensory signals can stimulate oxytocin release, influencing bonding, stress relief, and various affiliative behaviors. On the other hand, oxytocin itself can alter how we experience sensory input, especially pain, through its roles in both the central nervous system and the peripheral nervous system. These insights into the bidirectional relationship between oxytocin and somatosensory stimuli have substantial clinical relevance. For instance, therapies designed for autism, where social interaction can be challenging, might benefit from controlled sensory interventions combined with oxytocin-based treatments to enhance social bonding. Similarly, chronic pain conditions could be addressed by leveraging oxytocin's analgesic properties, possibly leading to more comprehensive pain management strategies.